we are going to switch over to the hands-on tutorial. So if you're just joining us, welcome to the Mobilize and Restore webinar series. And thanks to those who have continued on from the research talk. We're now going to switch over to the hands-on portion, the tutorial portion that will introduce pose estimation for biomechanics. And it will also be presented by Lukasz. And we'll take some more questions at the end of the tutorial, again, using the Q&A panel, and we look forward to more of your questions. And with that, I will let you take over again, Lukasz.
Perfect. Thank you again for the introduction and the invitation. And again, let me jump off and start off directly with the plan of our second part of the webinar. So there will be, we have around 15-20 minutes for the presentation slash tutorial part. So I'll go quickly through many points here, but you will get a link, and you already received a link to a notebook with all information, and it's kind of a self-contained lesson on how to use OpenPose that you can run without me talking. So that's a big valuable resource. So for now, I suggest you actually focus on just following what I'm showing, and then at the very end you'll have either time for asking questions or actually for playing around with this notebook and asking questions as you're exploring it. So we'll be working in a Google Colab, and Google Colab is an environment from Google that allows us to run any software on their cloud infrastructure without anything installed in your computer. So it will be extremely convenient for playing with OpenPose and with our sample scripts. I will show you how to download OpenPose to the environment, this environment, how to run a simple sample video on within this environment. How to interpret results that you get from OpenPose, and how to convert the output to a NumPy array, which is a way in Python to manage time series data or any matrix kind of scalar data. I will show you some pre-processing techniques. As I showed you earlier in the presentation, and as you may see already in this video, there's some noise in OpenPose. There are some missing observations due to occlusions. We'll try to deal with all those in the script. And finally, I will show you the analysis on how can we derive something meaningful from OpenPose results and how to export results to other environments. So the task that we'll be tackling is to go from a raw video collected with a single camera to go to the gate cycles and knee flexion in those gate cycles using OpenPose in the middle. So that's how Google Colab looks. That's, as I mentioned, that's an environment from Google that allows you to run any scripts, particularly in Python, and any software other than Python, like any binaries in the sandbox Google environment, so that you don't have to install anything on your computer. Here's a lot of text because we prepared a lot of explanation, but the main thing will be around running the code. So the Google Colab, you can think about that as your computer or any other computer. In particular, here we have a computer in the cloud, which you connect to through a web browser. And through this connection, you can interact with the computer, install software, and run our scripts. We'll be relying on OpenPose first. So we take the video, we run OpenPose to get the key points. And then as soon as we have those key points, we throw that into our Python programming scripts. We'll use NumPy for matrices, scikit-learn for some statistics and machine learning, and pandas for nicely displaying some of the data. So the main view of the Colab now, when we look at the code window, you can see that we have some text at the top with some nice headers that I prepared for you. We have some code blocks here that we run some scripts within, and then we get output of those scripts. So that's the main part that you will be interacting with, the main window. Now the second, to interact with that, you can add code pieces or you can add text pieces to make the notebook more readable for your collaborators, as we did here. And the other important view is this table of contents part on the navigator part, where we can display contents. If the notebook is divided into the section and subsections, this will nicely display in the table of contents. So pre-processing is somewhere in the middle here, as you can see. There are some other tabs in the navigator. You can search the web, search through your code. You can navigate help pages, and finally you can see the file system within the computer that we're in. So all the files will appear here as we download them or process them with OpenPose. And as you run your Google Colab, you're using some of the Google resources, so you see if you're short with RAM or with disk in your environment here. And if you are running out of resources, you can acquire some resources from Google, but that won't be necessary for this script. So for small scripts, you'll basically be able to run everything for free within this Colab environment. So let me show you how that actually looks like in practice. So let me switch the window now. I will run a few cells just to show you how to interact with that, but then we'll go back to the presentation to run it more smoothly. So as you will open the notebook, there's explanation of what we are doing and how to use it. There's a link to our paper here. And finally, we have one block for actually installing OpenPose. So you can press this play button and install OpenPose. It's already been installed in this environment, but like if you do it on your own copy, it will take 1 minute and OpenPose will be installed. Then we can download a video to run a cell with the code. We just press this play button or use the Control-Enter shortcut. Here's a lot of output, but what's important is that we are downloading from this link. We are downloading input MP4 file to input MP4 on your local drive. That will directly appear in the file system on the left. We can display the video with the play button. It shows the video that we downloaded. And finally, we can process it with OpenPose. So this takes around 18 seconds for this particular video. That's kind of an important part because like for all of your videos that we'll be processing, that looks exactly the same way. So let's wait those 18 seconds for this being processed. It's processed after 18 seconds. There's again a lot of output regarding some encoding of your video, but that's not relevant for our task. Let me just show you after clicking play how this processed video looks like, and that's what OpenPose outputs. So that's a beauty of Google Colab, that you are able to use the state-of-the-art open source deep learning project without installing anything on your computer. You're just interacting with this virtual environment and get all the output that is necessary for your downstream analysis using like on any laptop without any sophisticated hardware. You will be able to run state-of-the-art computationally heavy projects. So let's go back to the presentation. Now, I showed you how we can download a video with this widget function and the link to the video. So the link here was prepared specifically for this tutorial, but of course you can download any other video you like for processing in OpenPose. Those, after running OpenPose, as I showed you, you will get the output in your OpenPose directory. In the output directory, you have all the frames recorded. So I show you often the video of those key points recorded and embedded, hard-coded, so that we can visualize that. But what's important for the analysis are those more numeric outputs, which are JSON files describing positions of key points in every frame of the video. So that looks exactly like this for when we display one of the frames. So here the frame number 100. we load a so-called JSON format, which is basically a text file of this structure. And here I print this file to see that we have a version of OpenPose that was applied, and people as a list of people that OpenPose found in this given frame number 100. And the vector and the information that we are really interested in is this long vector of 75 numbers, the output of OpenPose for this frame. And there's 75 numbers because OpenPose detects 75 key points from 0 to 20. Sorry, it detects 25 key points and returns 3 numbers for each key point. So we have key points from 0 to 24, 25 key points. And for each key point, we have 3 numbers: X position from top left corner in pixels, Y position from the top left corner in pixels, and the confidence of OpenPose. So how sure OpenPose is that the key point is really in the place that it should be. And that's a numeric value from 0 to 1. To make it easier to deal with this information, we added some constant names. So key point number 0 is nose, key point number 1 is neck, key point number 24 is the right heel here. Now, those JSON files are semi-structured. Now, to really apply some signal processing, we need to write it in a more convenient format. So we wrote a small script that converts all those JSON files into a matrix where every row is one frame from the video. Every column corresponds to the number from this long vector of 20 or of 75 variables from OpenPose. So we have a matrix 344 frames by 75 columns, which correspond to the output of OpenPose. If we plot one of the signals, let's say we plot the position of the nose as a function of time or as a function of the frame, you will see this X coordinate like this, Y coordinate like this. And you already see that there are some problems with this output of the OpenPose. So you see that there are some zeros here, which look weirdly, and those are basically points that were not detected due to some occlusions. Nose might have not been visible, so we missed those points. But then we see that the signal is quite continuous, so we could just easily fill in those gaps, and that's what we'll be doing in this presentation, trying to post-process OpenPose output. So if we look at the right ankle, there are 3 problems that we'll be fixing here. So one is that I've just mentioned, those gaps in the video that just OpenPose returns 0. The other one is the noise. Around here, you can see that there is some high frequency noise, mainly due to the low resolution of OpenPose. And finally, we have this U shape. While we know that the nose should be roughly at the same Y level, you'll see that for most of the variables, we have this U shape of the signal. And that's the artifact of the fact that we collect these videos from this fixed position of the camera while the subject works back and forth. We rotate the camera and they're further and closer to the camera. As a result, we're getting this bias in the shape of the Y curve. So let's see how we can deal with all those problems. First, to interpolate missing points, we basically will find those missing points. We'll find where the OpenPose gave 0, and we'll try to interpolate in those points by using linear interpolation. In the SciPy package that I mentioned earlier, we have the sub package called interpolate that gives us an easy way to build a function to fill in those gaps that creates this method for filling in the points. We apply it to this time series and we get rid of those gaps, as you can see, as the output is in this from this block of code. The second problem we are dealing with is this high frequency noise. So you can see that there's noise here, but after smoothing it with Gaussian filter is just one of the methods to filter out your data. Basically, it takes the current point as an estimate for the current point. It takes the value from the current point, the points from the left and right, and averages them out to smooth it to the given point. And as a result, you'll get something like this. You'll get rid of this high frequency noise. And finally, to get rid of this U shape, we will normalize everything to a fixed length of a certain bone. So the rationale for that is that since the patient was getting further and closer to the camera, they were appearing smaller and bigger. If we scale them by one of the lengths, we'll be able to get a more consistent signal. And indeed, we took a femur bone, so the distance between the hip and the knee, and normalized the signal, divided the signal by the scaling factor at each frame. I won't go into details that, but that's essentially what is happening here. And as a result, you see that the Y coordinate is now way more consistent, and X coordinate is also slightly more consistent. So that's all pre-processing. Now we are interested in measuring knee flexion as a function of time in a gait cycle. So first, let's look at the knee flexion angle. For computing knee flexion, we'll just use very basic geometry. We'll compute the inner product between two vectors, the vector between the femur and tibia, and the inner product divided by normalized lengths, divided by lengths of those bones will actually give us a cosine of this angle. So using arc cosine, we are able to get back to the actual angle in radians. Now by converting those radians into the degrees, you will get the output in degrees. So we can compute that function for every frame and get knee flexion in every frame of the video, as you can see at the bottom. Now, the next step that we're interested in is to divide this time series into segments of gait cycles. And for this presentation, we just wanted to choose the simplest way to derive gait cycles, which is not the most accurate, but works reasonably well. We'll be looking at the distance between the toes in both legs, and the peak distance will tell us, will give us some event that we can refer to as a start or the end of the gait cycle. So we are now looking at the distance between left big toe and the right big toe. This changes over time, of course, looks like this. We'll be looking at peaks of those functions. And if we identify peaks with some peak detection algorithm that we borrowed from some MATLAB implementation, you'll be able to find this peak distance between the two. We mark those with vertical lines. So we can see that it performs as it was supposed to. That's all automatic on top of our time series of distance of toes. Now we can take those peaks and apply them to our knee flexion gait cycle, knee flexion angle over time. So now we segmented the knee flexion into gait cycles, and we'll be interested in plotting those 4 first gait cycles and the knee flexions in gait cycles in one plot, so that we can compare those 4 curves. So now we just cut it and correct the x-axis. So the reason for like this additional step will be that in gait cycles you'll have different numbers of frames. In one gait cycle you're going to have 27 frames. In another gait cycle you have 32 frames. So we want to map them on the same 0-1 scale, interpolate to create a function of time, so that we can project them on the same space, even though they actually have different time space. But now if we project it to 0 to 100%, we can plot them on the same plot. So that's what this function will be doing. And that's our result. And finally, at the very end of the notebook, you will have this nice one cell that exports what we've just created to a CSV file. So in the file system on the left, you see the trajectories CSV file that's already created with this one line here. So that we save our matrix as a CSV file and it lands in the file system from which you can, by clicking and downloading, you can get it now on your computer. So everything here was happening in the cloud, but you can download it to your computer. You can open it in Excel or LibreOffice or whatever spreadsheet tool you're using to see those time series as like in another software. Now you can port it to MATLAB, to R on your own computer and work with that downstream. So that's it. So that's the end of the tutorial. As I was mentioning, like everything was happening super quick because we are short with time. But I hope that you're going to go back to the recording, but also in the notebook itself, you can see all the explanations of all steps. We're trying to make it as precise and clear as possible. So hopefully we succeeded. If you have some comments on that, I will be happy to improve the notebook. And now we'll have a few minutes to actually test it out and answer some questions. Thank you.
Great, thanks again, Lukasz. So now we'll go ahead and switch back to the questions. We will focus on any that are specifically related to the tutorial, but if we get through all those, we can take a look at some of the other questions that come in. Let's see. So a question from Real Peng Sun: for spatial gate parameter extraction, do you need to perform any calibration with the image first to derive the distance measure from pixels?
So OpenPose is very robust and works on very diverse images. So it was trained on completely different images that we had in our videos, and it still performs very well in videos. So that works well without any pre-processing of images. But now for the output, to make it to feed that into our neural network that I was showing in the first part of the webinar, we actually applied all those pre-processing steps. And depending on what you want to do with those videos, you will very likely need some kind of normalization of your data, no matter if it's neural networks or some signal processing and direct pipelines. So, right, like you need a lot of post-processing after OpenPose, but not much before OpenPose.
Right. I mean, with a single video, I don't know if this helps clarify at all. You don't know the scaling of the person, but you can still extract the information about the movement of the segments relative to each other, which is what joint kinematics is in general.
Right. Okay. Yeah.
Okay. Let's see. A question from Gil Sarancoli. Out of curiosity, does OpenPose run faster in Google Colab than in a regular laptop, or any performance things to keep in mind?
So Google Colab gives you the simplest GPU possible. So it will perform worse than any hardware that you can get in a desktop computer, but like it will be much better than in your laptop, very likely. Like the GPUs that you get in laptops are slow, and very likely Google Colab will be better because you have this hardware acceleration. When you don't have a NVIDIA GPU, it takes 30 times longer. So instead of this 18 seconds that we had for OpenPose, it would take a couple of minutes or like up to an hour for a very short video on a laptop.
Okay, thanks, Lukasz. Now from Carla Musali, do you need some kind of affine transformation if, for example, the camera is filming from a different angle? Than the data was trained with.
For sure. For running our pipeline, since we didn't, like we had this question about adversarial approaches in the first part of the webinar, and like there are ways to make our models way more robust, but we didn't do any of that. So now, in order to use our models, you would need to bring your data to a format as similar as possible to what we trained our models on. So indeed, like if the camera angle is different, you would need some transformations or some retraining of algorithms for your data. Yeah.
Okay. So this question has come up a couple times, so I'll ask it. Do you know of any approaches similar to this looking at the upper extremity?
I haven't seen.
I couldn't think of anything. But.
Yeah, right. So OpenPose, of course, works right away for upper extremities, but then for using So what I showed in actual, the second part of webinar, you could do exactly the same for upper extremities, because that didn't rely on any data sets. For what I showed earlier in our research work, really relied on this data set of 2,000 videos, and I haven't seen a data set like this for upper extremity movements.
Okay. Now a question from Bram Vandenbosch. What's the difference between this workflow and that of DeepLabCut? What's different under the hood?
So DeepLabCut can give you similar key points, or even key points that you may be interested in more. So let me maybe say what DeepLabCut is. So DeepLabCut is kind of upstream from what I showed. So if you have a bunch of videos collected in your laboratory and you're interested in some key points in those videos, then you annotate some of those videos, and DeepLabCut allows you to train a model to replicate those manual annotations, and then you could run our workflow as well. So it's kind of complementary, not really competitive in this particular workflow.
Okay, thanks, Lukasz. Now a question from Kat Steele. Hi, Kat. Says, very cool. Thanks, Lukasz. How are you experimenting with integrating this with OpenSim? So
the main problem here, or main challenge or opportunity here, is that we have only 2D videos. Now, there are many 3D positions that can correspond to those 3D to those 2D projections. So with one video, it's quite challenging to get something as sophisticated as like dynamics. But if we have more videos, we could reconstruct 3D kinematics, and OpenSim could actually help us to denoise some of the data. So what we are trying to do is actually to use more cameras now and combine this 2D key point detection with some optimization that relies on OpenSim models. But like there's no direct way to do that. Like that will be our big next project.
Yeah, definitely. That's the big question. I have another big question. This is from Jason Moore. So he says the R squared of 0.51 seems to indicate that OpenPose is very poor at reproducing mocap data from a single video. Can you confirm that and give us an idea of what the best OpenPose can be with a single video? That's
a good question. So there are two sources of error. First is that we have 2D projection, and here the sagittal plane wasn't really sagittal. We were following the subject with the camera in the fixed positions. The subject was walking, so the angle is actually changing. So it's not really directly correlated to what mockup gives in the sagittal plane. So that's the first source of error. The second is that those key points were not really annotated by biomechanists. So people who were annotating those large data sets that OpenPose was trained on didn't know where the knee center, well, I mean, they knew where the knee center is as per their protocol, but not for what we are interested in biomechanics necessarily. So all that could be improved by annotating better data sets, by collecting better data. So I wouldn't say that the OpenPose is, yeah, well, it's a correct conclusion that the OpenPose is not that good with replicating motion capture, but that's that we showed that this can be denoised with some deep learning on top of that. And the OpenPose is not only tool here, so we could definitely build better tools, and that's the hope.
Right. Thank you, Lukasz. We have time for maybe a couple more questions, Lukasz. Are you?
Yeah,
you up
sure.
for a couple more? Okay.
Sure.
Let's see. Out of curiosity, are there possibilities to combine this video output with the IMU sensors in Google Colab?
Yes, so the Google Colab is whatever you can do on your computer, you can do in Google Colab. So that there's no question about that. For IMU, as soon as you have data, time series data from IMUs, And as we are creating this large data matrix, we could have more time series here. Sorry, that's actually not ready. But yes, we could just combine those signals in a big matrix and yeah, and do exactly what we're doing here. So as soon as you have data, you can combine. And as I was mentioning earlier, at CMU, Annie Halila and collaborators are actually collecting this kind of data sets. So I think they will be actually released and you can play with that quite soon.
Yeah, so definitely possible. I think you have to figure out as a researcher, data scientist, exactly how you're going to use the data, but definitely possible. Let's see, maybe we'll take one more question here before we wrap up. This is from Olivia Botanis, and I think a good question to wrap up. So what do you predict is the biggest challenge to moving such laboratory explored technology to a deployable smartphone application?
That's a great question. And like, that's something that we've been investigating for a lot. And we're actually explicitly running experiments to figure that out. So our thoughts so far is that the technology for some applications, maybe for simpler tests than walking, like for sit to stand test, which is common in physical therapy and in orthopedics and in assessing neurological disorders. Those technologies seem to be good enough for assessing some parameters of those simpler movements. So the question is more on the soft side, like, are people actually able to collect those videos in a good way? So when you ask elderly population, like some older subjects, to record themselves with the mobile phone, they can do that. Like, there's no doubt about that, but like positioning the camera in the right way, recording the 5-second video instead of like a very long video. Like, there are all those small technical soft questions and problems that need to be solved before we really use it in practice. So I think we are there or almost there on the technology side, but the question is how to actually make the user experience bearable and easy enough to actually make people commit to that.
Right, and maybe you touched on this a little bit, but I think the other part is making sure that the information that we're collecting is useful to patients, useful to clinicians, and they can actually act on it and have it improve their treatment or care, not getting just a bunch more data that, here's what your knee angle is without knowing exactly what to do with it. I think is another big challenge too.
Right. We try to test what we've built at home, and we asked patients to walk those 10 meters, and it turns out that in many apartments they don't have really 10 meters to walk freely. So it's not that easy as it sounds, but like, it's an interesting problem to solve. Like, and there are lots of stakeholders that need to be involved to actually make it useful for the humanity.
Yeah, but that's our goal, and I think it'll be a team effort from the research community to tackle all of that. So we're excited to have so many of you join the webinar today and ask great questions. So we'll go ahead and wrap up here. Thank you again, Lukasz, for a great talk. Thank you to everyone for all the great questions. There'll be a survey that will appear in the pop-up window, I think, at the conclusion of the webinar. Please fill that out. That'll help us improve the webinar series and choose upcoming topics. This webinar series is supported by 2 grants from the NIH, including the grants that support our Mobilize Center and Restore Center. We're grateful for the support, which allows us to run the webinars and other events. Thank you everyone for participating. We hope you'll continue to stay involved with our centers and that we'll see you at a future webinar. See everyone. Thanks again, Lukasz.
Thank you, everyone. Bye.